Okay, so before we start, let's think a little bit. How would you define omics data? You've probably heard of genomics, transcriptomics, maybe phenomics, proteomics, etc. So there's many different types of omics data. So how would you define what is omics data? What is common between these different omics types of data? And another question that I would like you to consider is if you can think of any example of big data in healthcare. Maybe you're thinking electronic medical records or devices sending signals, and that becomes a large data source. What other examples would you have of big data in healthcare? So the title of the talk today is Personalized Medicine, How We Can Improve Healthcare and Reduce Costs of this personalized approach through many different sectors across research and discovery, education, analysis of the data, maybe industry, pharma industry, healthcare industry, etc. So how we can improve the delivery of and how can we reduce the costs in personalized medicine. So again, think for a moment, what is personalized medicine and how is that different from non-personalized medicine or evidence-based medicine? Have you thought of any examples of personalized medicine or maybe reasons for non-personal medicine? And again, if you think about this, personalized medicine, of course, is an overused term, somewhat of a cliche. And we also tend to call it precision medicine, stratified medicine, various terminologies. For this presentation, we're just going to stick to personalized medicine, and we'll see how it goes. So what is the problem? The problem is that healthcare is not personal and not precise. A lot of times we have imprecise measurements. There's only a few options. There's a lot of lookalike diagnostics, right? So we see a symptom, it looks like something else, and we go from there. So there's a lot of analogies, and then try and see treatment. So let's try this treatment for a couple of weeks. If it doesn't work, we'll try something else. And of course, as you know, it's a good example is the resistance to antibiotics that is developing because of this try and see treatment. And bottom line, it's leading to bad outcomes. Especially we can see this, for example, in cancer epidemic. Right? So 1.7 million new cases of cancer will be diagnosed in the U.S. This year, and about 600,000 people will die from cancer in the U.S. alone. And what's surprising is that about 40% of the total population will be diagnosed with cancer at some point during their lifetimes. And 40% is close to a half, which is a lot of people. Just to drive this point home, 3 new cases and 1 death per minute. So we've been doing this for a few minutes. You can think about how many people have been diagnosed or died from cancer during this time. So the 2 main components of personalized medicine are diagnostics and therapeutics, right? So how do you diagnose a condition in a more personal, precise way? And how do you then treat it in a more personal and precise way? So on the diagnostics, for example, we see that about 40,000 cancer patients die each year as a result of misdiagnosis or delayed diagnosis. This is an interesting statistic, but also you can see that diagnostics plays a critical role. And so why is it so complicated? So it's complicated because cancer is so complex. So what we call cancer is actually many different diseases with many different types and subtypes. And what you can see here is a chart that you can find online called OncoTree. And you can see how many different types and subtypes and subtypes of subtypes of breast cancer we see on this chart. So what is the need for molecular precision in this case? Well, to identify precisely the type of a cancer, you really need to know some of the genetic differences. So you can see the 2 different pictures of different cancers, and you can see how the visual picture is different, but of course also the genetic component of why those morphological differences occur. So really it's a dysregulation of gene expression that leads to cancer and defines cancer subtype. For a second, let's just think about the value of diagnostics. So diagnostics, we started kind of in cancer, but of course it's a much broader topic. Only about 2% of worldwide healthcare spending is used on diagnostics, but the benefits is about 70% of the conditions, and 70% of the decision making is influenced by diagnostics. So it's a critical component of healthcare. You can see that it applies to many different conditions, including cardiovascular, personalized medicine, cancer, point of care, infectious diseases. And we also have here on the side a lady that suffered cancer early on, actually at the age of 25. And because she had precision diagnostics and a team that knew how to utilize those diagnostics, she is now healthy, completely healthy. So what about the cost of diagnostics, molecular diagnostics? Well, molecular data since 2014 has been widely available, and the cost to generate the data has been below $1,000, which is approximately the cost of generating the CT scan data. And now we are even below the $460 cost of X-rays. And Illumina just recently announced a new machine that will provide sequencing data for less than $100 per sample. All right, so the second major component of healthcare, of course, is therapeutics. And here we have a good stat. About $350 billion was wasted on prescription drugs in 2011. Okay, let's return to our cancer case and think, here's an example of a $150,000 per year drug called Opdivo. It's an immunotherapy therapy for lung cancer, and immunotherapy, of course, implies a more personalized approach. But here we also have this other statistic that on average, the median improvement in survival for 71 drugs approved by the U.S. Food and Drug Administration for treating solid cancers over the last decade, the improvement is only 2.1 months. So really it extends your life by about 2 months. And that's not a big improvement. And also 75% of cancer drugs have no effect on patients. So now, before we go on, let's think about it for a few minutes. What are some reasons for these drugs being ineffective? Is it the design? Is it the delivery? Is it the patient adherence? So many different factors here, of course. What are some of the reasons for cancer drug costs? Why does it cost so much to develop this? Why does it cost so much to bring it to market, to get it approved? And also, what are the reasons for cancer care being so costly, right? So it's not just therapeutics, but cancer care in general. All right, so here's a good example of a real-world scenario where you have similar clinical conditions, but they differ in their molecular features. So, for example, you have immunohistochemistry or FISH test for a patient, and the patient gets identified as a HER2-mu breast cancer patient and gets prescribed with this treatment, Herceptin. And what we know today is that a portion of those patients actually do not respond to Herceptin at all. Why? Well, because using the PAM50 molecular test, which is a test that contains gene expression of about 50 genes, 50 genes called PAM50, will be identified as the luminal subtype, and those patients do not respond to treatment. So think about this. Think about the changing economics of delivering this kind of care. So we have this lung cancer subgroup that is called ALK positive non-squamous non-small cell lung cancer, NSCLC. These patients have been responding to a new treatment that's coming to market, and they respond well. So you can see that a proportion of them actually respond really well to treatment. But the problem is that there's only 13,000 of those people out of 14 million people diagnosed with lung cancer. So it's less than 1%. So think about the marketing, the development, the research, the marketing efforts, the delivery efforts to identify those right patients and to deliver them this kind of care. But in an ideal world, if we had great diagnostics and great therapeutics, what we would see, of course, is that a number of different tests would help the clinicians match the patient with the right treatment, know their risk, and match them to clinical trials if the clinical trial is available. All right, so let's look at the process of delivery. How does this work? So a patient comes into a doctor, a clinician. What they get referred to is a clinical lab. That clinical lab has to be able to generate the right type of data, take the right type of sample, of course, generate the right type of data, perform analysis of that data, and then interpret that data, right? So the clinician, after the patient comes back, has to be interpreting that data correctly. So that's where we see the data component, right? If you think of the type of data that we're dealing with, that is the big data, the big omics data that we will have. Let's look at all the different things that describe molecular data. So we have DNA mutation variants. We have expression analysis, non-coding RNA, RNA editing events, transcriptome, different transcriptomes here, ChIP-seq and bisulfite-seq, epigenetic regulation. We also have mass spec data like metabolomics and proteomics and lipidomics. And all of this data speaks about these biological events. And to interpret those biological events, you have statistical analysis of the data, integration of that data, and then you have machine learning. And that is really what we do at Pine Biotech. All right, so what do you think? Who is the user of molecular data? What are some barriers to the use of molecular data? And how can we be more effective in the analysis of data? Okay, so now what I want to do is just continue speaking a little bit about TBio, our discovery platform that is focused on delivering high-speed analysis and visualization tools so that people can work with multi-omics data and integrate that data and also learn from these projects. So what is our vision? Well, in the 1950s, the DNA double helix structure was discovered. And throughout the 1980s, the genetic code was cracked and the first DNA sequencing technology was developed. Eventually, by 2015 and 16, we see proliferation of data that becomes widely available throughout research and eventually in healthcare. Furthermore, what we see is that in the near future, we will see more and more of this data being utilized in healthcare. So what we see is our role that we want to play is analysis, integration of data to provide diagnostics and stratification. First of all, for precision diagnostics, and eventually to support personalized medical treatment selection. So why is this so complex? So it's so complex because we have different networks, different levels of data, organ networks, cellular networks, molecular networks, and genes. And all of these networks actually interact in different ways. And so genes and their regulation, that is more of the omics data today, but more of the organ networks, that is the clinical data, right? Those are the data sets studied in a clinical setting. But we see more and more merging of those two types of data sets and omics data crossing over into the clinical data. Multi-omics. Multi-omics is a concept of many different types of omics data phenomics, such as cell growth, for example, or cell shape, genomics, for example, single nucleotide variations, SNV or SNP, polymorphisms, epigenomics, chromatin remodeling, for example, transcriptomics, alternative splicing events, repeatomics, transposons, proteomics, how genes are expressed and turned into proteins, and eventually metabolomics, metagenomics, and even other components like the microbiome, the virome. And so all of these data, right, they play together, they affect they affect one another. And to understand the underlying biological processes, we need to see all of those data points together. What are we working on? Well, we are working on really a lot of different areas, not just personalized medicine. We have projects in virology, in drug design, in biomarker discovery, in epidemiology with many different institutions. As an example, I want to give you a practical project where if we look at stage 4 castrate-resistant prostate cancer, you can see the challenge of treatment selection in combination with chemo, determining length of treatment, maybe a combination of different treatments, and assigning patients to match with clinical trials. And this is kind of like provides you a translation of research project of integration of different features of data, integrating them into the clinical decision support for pharma diagnostics and healthcare. Okay, so as I mentioned, we have this platform. This platform works right now for research projects and education. We're actually coming out of beta, and we've tested this on a number of validation projects in collaboration with various researchers, and we've gotten some great feedback, as you can see, on the different types of data sets that we've analyzed and the tools that we provide. All right, well, thank you for joining me today and hearing about personalized medicine and what Pi Biotech is doing in personalized medicine. I'm excited to participate in the work that our team does, and in the next meeting we will start going over some of the projects that we've been working on, some examples of how this data gets integrated and analyzed, the platform itself, some of the tools that we have, as well as some of the specific collaborations that we would like to discuss. Thank you.